Okay, so good afternoon. I think there are a couple of students who have not yet mailed their answer sheets. Okay, I counted and I saw only like 21 or so. So I think two of them have not mailed this thing. I'll cross check and then I will let you know. Okay. People are joining. So we were looking at the antisense technology and also the RNA interference technology, right? The antisense technology is very simple, straightforward. Basically involves taking the complementary strand of the messenger RNA and introducing somehow introduce the complementary message into the cell. So in reality, it will go and specifically block only that particular messenger RNA, right? Everything else looks fine, looks same. And so ideally, it is going to be very specific to the cells that or the messenger RNA that we are targeting. Well, it turned out that introducing the silencing antisense RNA turned out to be a major challenge. Okay, so it was not a very successful thing. Although things are changing, and it is likely that the antisense drug candidate can come out as early as in the next few decades, less than a decade. Okay. Typically, if you look at any drug discovery, it takes about 14 years. From the time the process starts. Suppose somebody puts out $1 million to you guys and say, okay, class of 5th semester BSc, let's do this together, and let's start a program. And if you were to start a drug discovery program in 2021, it's going to be good 20 to 15 years before it comes out into the market. So it's a long drawn out procedure. Okay. So it turns out that delivery into specific cells was an issue. And then people somehow I won't say forgot, but antisense technology was pretty much people are figuring out how to do this better, right? And then it turns out that RNAi came. RNAi was something that was totally unexpected. Just introducing a double-stranded RNA or a hairpin-like structure within an RNA resulted in the degradation of the endogenous messenger RNA. People were surprised, and when they figured out the mechanism, they got a Nobel Prize. It turns out that this work was already done in plants. This was established in plants. It was called as post-transcriptional gene silencing in plants. This was also called as quelling, Quelling, quelling in Neurospora crassa and few more fungal species. But then it turns out that this process was called RNAi, and it got stuck, and then RNAi it turns out to be a very powerful tool to introduce, to do what is called as gene silencing. You're essentially trying to silence a gene. It could be one gene, it could be multiple genes, but essentially you are trying to, correct this thing. It's possible that there are certain genetic diseases which are monogenic. That means the underlying cause is just because of one single faulty gene. Okay, if somehow you could stop that gene from functioning, you could in theory, cure the disease, right? Some are polygenic diseases. For example, cancer is typically a polygenic disease. Many things have gone wrong, and the cells are hurtling toward a path of this thing, uncontrolled division. And so it is a little tough. But then I think what people are realizing is that each cancer is also unique. For example, you may have hundreds of lung cancer patients. Each of them carries a unique mutation, and that is why, you know, even though within lung cancer, you may give people, some people respond, some people do not respond. Okay, so it turns out that it could basically be the genetics of the patients, right? That could be responsible for the patient being responsive to treatment or being not responsive to treatment. So I just saw this video from my favorite person, Professor Eric Lander. So let's have a quick look at that video before we go into the remainder of the class. Okay.
And it makes a messenger RNA. Here's my messenger RNA. And instead of destroying my gene by a mutation, suppose I could somehow destroy just the messenger RNAs associated with the gene. Specifically target the messenger RNAs for a specific gene. That would be kind of cool, because if I can make sure the gene doesn't produce any messenger RNA, it's not going to make any protein. It's like having a mutant. It's a mutant without being a mutant. How am I going to do that? Well, people said, this isn't that hard. They said, what we're going to do is we're going to design a complementary piece of RNA that will bind to my messenger RNA of interest and prevent it from being translated. Antisense RNA. It was so simple, so clever. They were going to put in a strand of RNA that matched your RNA. It would bind to it and block it. Ridiculously simple, isn't it? And you can make it for any RNA you wanted.
I hope it is audible.
Yes, sir.
The problem is, it didn't work very well. Most of these pieces of antisense RNA, when you injected them into the cell, did a pretty crummy job of blocking the RNA, because, you know, you need a lot of them to bind to that RNA. You know, it just didn't work very well. And then, as sometimes happens, a wonderfully serendipitous experiment occurred. Some folks, Andy Fire and Craig Mello, were working on this beautiful genetic organism, the nematode worm called C. elegans, a very elegant, sinuous, little tiny round worm like this. And what they were doing is they were injecting antisense RNA against a particular gene, a gene called twitcher, which when mutated caused the worm to twitch. Mm-hmm. So what they did was they injected, here's the RNA they wanted to hit. They injected an antisense RNA. And they really didn't get much. They also, by the way, just as a control, here's the RNA that they were trying to hit, mRNA. This is the mRNA. This is the worm, by the way. They injected sense RNA. That is a little strand of RNA of the same length that was not complementary to the messenger, but was the same as the messenger. And that wouldn't bind to the messenger, would it? No. And did that have much effect? No, didn't have much effect. So neither the antisense nor the sense really was very effective. And then they did the following ridiculous control. They took the worm
Okay, so since morning, there has been this network disturbance. So every now and then, it may stop, but don't worry about it. We will continue with our class. Okay.
And both alone, each alone does nothing, really, right? The antisense turned out not to be very effective despite it being a good idea. The sense doesn't even bind to it. And of course, since each alone does nothing, together, they, of course, caused it to twitch. It was incredibly effective at destroying the RNA from that gene. Somehow, double-stranded RNA was a very powerful way of destroying RNA. This was not the expected result. This also, by the way, won a Nobel Prize. Let me tell you why. Because it turns out it revealed an amazing natural mechanism in cells about which we had known nothing. It turns out that when there is double-stranded RNA in these cells, double-stranded RNA gets cut down to about 21 base pairs, chopped up to about 21 base pairs. So first you dice it up into little pieces. It's diced up into pieces of about 21 base pairs by an enzyme called dicer. And then it gets loaded into another protein. One of those strands gets loaded into another protein, just one of those strands, and it becomes a template to go destroy any matching RNA. And you actually have a natural mechanism that does this. This is called the RNA-induced silencing complex. RNA-induced silencing complex, or RISC. And what happens is this complex, the RISC complex, loaded with this 21 base pair RNA, goes off looking for messages that match it. And it will destroy those messages. Got it? Add a double-stranded RNA. It will then go off and kill in a catalytic fashion, reusing that template again and again and again and again to destroy matching messages. Why do you have such a nutty mechanism? Viruses, double-stranded RNA viruses exist. Normally, you're not supposed to have double-stranded RNA, are you? Messages are single-stranded. Double-stranded RNA should be a red bells, red alarm bells. Something's wrong. Virus, virus, virus. There's a defense mechanism against double-stranded viruses, double-stranded RNA viruses, that cuts and then uses the stuff that's been cut out to destroy virus. That's probably the way it works in plants and a number of organisms. In mammals, it probably has another very important function. Remember we talked about transposons? Transposons are making copies of themselves all over the place. There's so many copies of transposon RNAs. Some of them will be on one strand, some of them on the other strand. Anything that's highly repeated in the genome will likely give rise to matching messenger RNAs. And it can be used to control transposons. Transposons are, of course, an internal virus, in effect, in a way. And probably in us, it's playing more of that role of keeping transposons in check. But basically, double-stranded RNAs are not supposed to be running around, whether it's coming from a virus or coming from too many copies of transposons from each strand and all. And this mechanism keeps them in check. It's very powerful. It's an ancient defense mechanism. We now can subvert that ancient defense mechanism that was accidentally stumbled upon in the C. elegans experiment here, with the injecting and getting the twitching worm there. And all I have to do is take a cell and put in double-stranded RNA. You can buy these inhibitory RNAs for every gene in the genome. In fact, you can make viruses that carry, here's a viral RNA. Certain viruses, lentiviruses, you can make them to have these 21 base pair matching double-stranded sequences. Put the virus into the cell. The virus will then, when it makes here an RNA, that'll be processed down into 21 base pairs, and it will be an inhibitor of those genes. And people have made, in fact, we here at MIT have made libraries of inhibitory RNAs against every gene in the genome for human and for mouse. So we can, in fact, add these back to cells and knock down genes. And you can knock down thousands of genes in different wells of a plate. Now, it turns out this is pretty powerful. I don't have to make knockout mice. Of course, I don't get mice. I'm just working with cells. And it's not perfect. Unlike knocking out the gene, knocking down the RNA is not 100%. I can knock it down a lot, but I often can't get it down to 0. And sometimes it doesn't perfectly knock down its own RNA without knocking down some other things. So they're a little messy. They're quick and dirty, but very powerful. Gold standard would be a mouse knockout, but a lot of work for each of those. But I can also knock out thousands of genes in parallel using, or knock down thousands of genes in parallel using RNAi, RNA interference, and one can do that as well. So these are two very cool tools. Now, this we're using routinely in all the labs these days. RNA interference, very powerful method. Okay, stop for a moment and answer this question about RNA interference. I'm going to close by talking about two cool new methods for engineering genomes, editing genomes, and they're pretty cool. So hang on for section 3. By modern genome editing, I mean that one of the things I'm going to tell you about has really only been being used for the last 3 years, maybe. The other one has only been used for the last year. All right, that's what modern means in molecular biology. And I'll just be brief about them. It turns out, as happens with everything we're doing, there's something weird somewhere in life. We figure it out, and it becomes a tool for us. So there's something weird. There's a particular plant pathogen called Xanthomonas. So Xanthomonas is a plant pathogen. It's a bacterial plant pathogen. And what it does is it injects into plant cells a protein. It's a protein that binds to DNA and activates genes in the plant. It kind of takes over the control program of the plant by injecting a protein that knows how to bind to DNA and activate certain genes. Like I was talking about, I've talked about before how the LAC repressor bound in certain places or the estrogen receptor bound in certain places. Suppose this plant was smart enough to make a protein that bound to a particular set of genes it wanted to turn on, and it could turn them on. Well, it did that. It has certain proteins, and this class of proteins goes by the name talons. These talon proteins can target things. They are transcriptional activators of genes, and they can target things. And I showed you very early in the course a cool protein that could wrap around the DNA double helix. Very early on when we talked about protein structure, I showed you this guy. It's wrapping around and around the double helix. These are talons. They can bind to DNA. Every one of these subunits specifies one base of DNA in the sequence it's going to bind to. And it turns out to be a beautifully general code. There is a 34 amino acid unit of which there can be many copies. This 34 amino acid repeat unit comes in 4 flavors. Flavor red binds T. Flavor green binds A. This one binds C. That one binds G. They're identical except for these middle two amino acids. You change the middle two amino acids, you can either bind T, C, A, or G. So suppose I wanted to make a protein that bound the sequence AAGT, GC, AC, GT, A. I just look up the sequence. I say, it's this subunit, that subunit, that subunit, da, 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 da. I can just write the protein. I can order up any protein, I can order up a protein to bind any given sequence of the genome I'm looking for. We did not think this up. Xanthomonas thought this up. Xanthomonas uses this in different ways to bind different genes. But once we know, we can make our own proteins. If I want to make a protein that binds any particular spot in the genome, I just string together these TALEN subunits, these 34 amino acid repeats by choosing the right ones, and I can target any gene in the genome. So, suppose there was a particular gene that I wanted to activate. I could build a TALEN that bound exactly to that sequence. Oh, and maybe also had attached to it an activator domain that helped attract the RNA polymerase. Or maybe I want to repress any gene in the genome. I could pick that gene in the genome. I could fashion me a TALEN that bound there. And I could attach a repressor domain that would help repress transcription. Suppose I wanted to cut any gene in the genome, just snip it right at a particular location. Turns out, I can take that location in the genome, make a TALEN that binds here, and attach to it half of a restriction enzyme dimer. And I can make a TALEN that binds here and attach to it the other half of a restriction enzyme dimer. Only at the place where those two proteins sit down next to each other does the dimer form, and then it cuts the DNA there. So I can edit the genome. When it gets cut, what happens is it's cut, enzymes come along madly, chew it back a little bit, slam it back together, but it's almost always a broken gene. So I can take these TALENs and I can attach things that are transcriptional activators or repressors at that one location. I can attach this particular nuclease and cut at those locations. I can direct whatever I want to, wherever I want in the genome. And in the past year or two, people have come up with very efficient ways to build TALENs on demand and build 100 different TALENs in a few days to target different sequences in the genome. And it's pretty cool. Yes?
So these subunits are protein, not any nucleic acid.
Sorry? Those TALENs?
The colored units.
Those are proteins. Those are proteins. They're 34 amino acids. This whole set of 34 amino acids is forming a whole domain whose entire purpose is to recognize one letter of DNA. Let's go back to that picture. Let's go back to, it's mind-blowingly cool, right? Let's go back to that picture. This guy is wrapping around. Here's one of those 34 amino acid things, and it's making contact with one nucleotide. Here's the next 34 amino acid thing. It's making contact with one nucleotide. Here's the next 34 amino acid thing, making contact with one base of the DNA. And it's just wrapping around and reading the DNA as it's going.
How do you get that big of a protein inside it?
How do you get that big of a protein inside a cell? You put the gene for it inside of the cell. You transfer the gene, let's say, by transfecting it in or with a virus, and you make it inside of the cell. That's how. You just make everything inside the cell. You don't get it in there any other way. All right, and they're very pretty too, in addition to being way cool. All right, one last trick. This is the one, by the way, that there was a popular demand for a little bit earlier. CRISPR. Last cool system I'm going to tell you about, CRISPR. Yet another shaggy dog story about something that goes on in the cell. Turns out that bacteria, lots of bacteria, maybe 40% of bacteria, have a very funny region in their genome. Just a weird region in their genome. They have a cluster of palindromic repeats, regularly spaced, repeated sequences in the DNA that are regularly spaced, clustered, regular, short palindromic repeats. And then there's just stuff in between. And, everybody said, kind of weird that bacteria have these repetitive things interrupted by equal size spacers. And of course, the people were mostly thinking about these repetitive things and not thinking about the spacers. This went on for quite a long time because these things were present in many bacteria, but the spacers were considered just spacers. The secret was the spacers. Turns out those spacers match bacterial viruses. This might match virus number 22. And that guy might match a piece of virus number 37. I'm just making up their names here. But what was eventually realized is these spacers matched viruses that might infect the bacteria. What they were a little library of information of segments of viruses that might infect you if you were that bacteria. It was an immune memory. It was a memory system of viruses that had previously infected the ancestor of that cell. And it kept that memory stored in its DNA in a very explicit fashion. And what it would do is it would make RNA. It would make an RNA from this CRISPR locus. The RNA would then get processed up into separate pieces. And each of those pieces, processed up like that, could then be used by a certain protein present in the bacteria to search and destroy any matching DNA. It would cut any matching DNA. So just as your body has immune cells that are able to recognize invaders you've seen before, viruses you've seen before, the bacteria has stored in its genome a record of viruses that were seen before. And it's constantly making these RNAs, chopping them up, and those RNAs allow you to cut a matching piece of DNA. And all you have to do is specify about 20-odd bases. These are about 20-odd bases long, and it will direct and cut the matching piece of DNA. Insidiously clever. Now, of course, when a virus gets infected with a virus, when a bacteria gets infected with a virus, often the bacteria will die. But sometimes the bacteria lives, and it's those bacteria that capture pieces of the virus and stick it into their CRISPR locus, their CRISPR repository, their little CRISPR file of things that our species has seen before, and uses them to kill DNA, or to cut DNA. But of course, now that we know how it works, we can make our own CRISPR. Suppose you want to cut a particular human gene, a particular mouse gene. Just make a CRISPR. Make a CRISPR RNA that has that little sequence and has those little flanking things, and add the one protein, it goes by the funny name Cas9, add the Cas9 protein together with a little set of CRISPR instructions, and you can then cut at that gene. The ability to do this in mammalian cells is less than 12 months old. It's only just been developed. And everybody I know is off using CRISPRs to go edit the genome in all different ways and make cuts and make changes because it's incredibly quick. They're still working on all the details, how perfectly it has to match. A new paper just came out the other week about that. One that I just saw was being submitted on the details of how to perfectly design CRISPRs and things like that. But it's a pretty powerful tool. Any questions? There should be, because I may not even know the answers, because this is all still being discovered as we teach introductory biology. Yes? Why doesn't the CRISPR cut its own locus? That's a great question. I don't know. See, I'm not embarrassed to say for new stuff that I've never taught before. I don't know, actually. It's a great question. Why doesn't the CRISPR cut its own locus? It's supposed to cut DNA. I owe you an answer to that. Think maybe the little triangles will do it? Could be. Those triangles are, of course, just DNA sequences, not necessarily triangles. There aren't really triangles there. I just draw triangles. I'm not positive. There's got to be something, I'm sure, that'll protect the CRISPR locus. But if you remind me, I'm going to go find my colleague, Feng Zhang, who works on CRISPR, and we're going to find out. That's a great question. Why doesn't CRISPR cut its own locus? What else do I not know? Any other questions? Yes? Where's Cas9? It's encoded by another gene. So if I want to make this work in mammalian cells, I have to make a mammalian cell that has a Cas9 gene in it. So I just add back the Cas9 gene. But you guys are inured to that by now. You say, big deal. I'm just going to encode. I'm going to make a transgenic Cas9 protein in the cell. Yes. The CRISPR DNA locus is DNA, but it makes an RNA, and it's the RNA which is the active element that is used by the Cas9 protein to cut the DNA. It functions as a little RNA piece, but it's kept permanently in the DNA file of the DNA chromosome of the bacteria. Yes?
I was just going to ask, I think you kind of said it, but like the RNA is like held by the protein and that binds to the DNA? Right?
The RNA is held by the protein and binds. It's used as a guide to know what to cut. Pretty cool. All right. And I'm glad to be telling you about something that's just discovered, because it gives you the right sense that this is a totally fast-moving field. You're pretty much now up at the cutting edge of how we do things like genome editing. So what have we done? We have now closed the triangle. Given genes, we can mess around with the gene and its DNA. We can mess around with the gene and its RNA. Given a protein, we could find the gene and mess around with that gene. So I'm going to say we get credit for being able to go back from a protein by going to its gene and going back. We can now edit the genome. There are a lot of tools for editing the genome and make the genome of your choice there with edits, knocking out at the DNA level, knocking down at the RNA level, doing all sorts of different kinds of editing, even beginning to rewire cells, changing the wiring diagrams by adding talons that bind to particular places and bring particular activator domains. You have only begun to see what's going to happen with this as people can write their own circuitry in DNA. That completes our triangle, and that's all for today.
Okay. So I hope the class enjoyed this lecture. I think all the lectures by Professor Eric Lander are very, very interesting, right? And we have heard a couple of them previously. So this thing of gene editing, whether you use RNAi, whether you use TALENs or CRISPR-Cas, is that you now have the ability to correct genetic mistakes. For example, we all know that hemophilia is caused by a single gene, single letter, single nucleotide being changed, right? Now you have the ability to go back, correct it, and you can, check for the correctness, and then you can reintroduce that. So you now have the ability to cure genetic disease. What was deemed as incurable, right, are now curable. Of course, I think we are only looking at monogenic disease. That means diseases caused by mutations in a single gene, right? Very similar to this is another disease called SCID, which is severe combined immunodeficiency. That again happens because of a rare point mutation. You can go back and fix the adenosine deaminase gene. It's called ADA gene. You can fix the mutation and you can get back functional adenosine deaminase, and then you will recover from this SCID phenotype. All these things have been done in mouse. Okay, many of these experiments have been very successful in mouse. It is going to take time to translate these findings from mouse to human beings. It is going to take some more time, right? We all know that people who did the CRISPR-Cas, they won the Nobel Prize last year, right? So as Professor Eric Lander said, making sense or making meaning out of mundane observations, right? Serendipity, right? Making sense out of serendipity is what, earns people their Nobel, right? It is something that they have observed. People have known this and people didn't pay attention to it, but then somebody dug deep and found something that is profound, something that has applications, and it is able to change the way we do biology, right? Genetic engineering, to a large extent, has progressed based on these, small discoveries, but then their applications can be astounding. Okay. Any questions? Although CRISPR-Cas and TALENs are not part of the syllabus, I just thought these are technologies that are going to transform, the gene editing technologies. TALENs, of course, is not being used anymore because RNAi is much, RNAi and CRISPR-Cas are much easier, right? So people have moved. Even RNA is not being used as much as CRISPR-Cas is being used. Okay. Okay, so we will look at two short videos on RNAi. There is a company called Alnylam Therapeutics, which is in US. Okay, they have taken a major lead. I think there are several other companies, but they have taken a major lead in bringing therapeutics, RNAi-based therapeutics into the clinic. So we'll have a quick look at two short videos on this, and then we will take some questions if there are any questions.
If you want to achieve better results, make a great impression, and save time at work, you should get Grammarly.
With Grammarly, it's like I have my own personal editor. On every email or message or essay that
RNA interference, or RNAi, is a natural process of gene silencing that regulates gene expression. The discovery of RNAi was a revolutionary breakthrough and was awarded the 2006 Nobel Prize for Physiology or Medicine. Gene transcription begins in the cell nucleus, where messenger RNA, or mRNA, is produced from a double-stranded DNA template. The mRNA then travels into the cytoplasm, where ribosomes translate mRNA into proteins. Proteins mediate all cellular processes in both the normal and disease setting, with certain proteins involved in the cause or pathway of disease. To date, disease-causing proteins have been the target of conventional therapies, such as small molecules and antibodies. The RNAi mechanism enables the transient and selective silencing of any gene by targeting mRNA for degradation, hence preventing the expression of proteins and their function in disease. Alnylam is developing a potential new class of therapeutics, harnessing the function of small interfering RNA, or siRNA, the molecules that mediate RNAi. By chemically synthesizing and administering gene-specific siRNA molecules, Alnylam can harness the natural RNAi process to direct gene silencing, potentially representing a new frontier for treating disease. An siRNA is a short oligonucleotide duplex with a sequence complementary to the target mRNA. An siRNA forms a protein-RNA complex called the RNA-induced silencing complex, RISC, causing the siRNA to unwind, leading to the complementary functional strand being loaded into RISC. The sequence of the functional strand of siRNA directs the RISC complex to the targeted mRNA. The RISC complex degrades the mRNA, stopping the synthesis of the specific protein encoded by that mRNA, reducing unwanted gene expression. Furthermore, the functional strand-loaded RISC can target more copies of the same mRNA and destroy them. Thus, the actions of an siRNA are catalytic. RNA interference is a novel and innovative approach to specifically target and silence any gene that drives disease, including those that can't be targeted by other, more conventional therapeutic approaches. Alnylam is leading the translation of RNAi into an innovative new class of medicines. Our robust pipeline of investigational RNAi therapeutics is focused on diseases with high unmet medical need that fall into 4 strategic therapeutic areas: genetic diseases, cardiometabolic disease, hepatic infectious diseases, and CNS and ocular diseases.
There is this another company called Silence Therapeutics. Okay, we'll see a short video from that.
DNA, deoxyribonucleic acid. It's the genetic material that's in the cells of all living organisms. It's the double-stranded helix structure in cell nuclei that carries the genetic information of an organism. It consists of thousands of genes that are made up
This is based on the applications of RNA. Okay.
of millions and billions of pairs of nucleotides. Each gene is a set of instructions on how to build a protein molecule. But in order to make a protein from DNA, there's another player, messenger RNA. DNA is copied into mRNA, RNA, ribonucleic acid, a single-stranded sort of mirror image temporary version of DNA. This mRNA transfers the DNA message from the nucleus to the ribosomes, where the proteins are manufactured. The mRNA is then translated into proteins. And in turn, these proteins do things. They are the workhorses for cellular and organism function. Taken together, DNA, RNA, and proteins are the blueprints and raw materials for life. Knowing that these molecules exist in living organisms and how they work gives humans some powerful tools for manipulating the genetics of organisms. Well, this brings up another molecule involved. It's a version of RNA, that temporary copy of the DNA, called RNAi. The I in RNAi stands for interference. When a cell wants to stop making a protein, it produces a little RNAi molecule which silences certain DNA from producing a protein. RNAi is a naturally occurring, necessary genetic component of all organisms, including humans. For example, RNAi has the important job of fighting things like viruses and regulating genetic changes and mutations. RNAi does this by specifically targeting certain sequences of DNA and blocking the production of proteins. Since the discovery of the RNAi process in the 1990s, this genetic mechanism has led to some pretty innovative applications. Application of RNAi has shown to be a promising method of improving life for us on many levels, including switching off genes that cause diseases, learning what genes do and how they work, and making food production easier. Researchers use RNAi by designing and introducing short strands of RNA, around 21 to 25 nucleotides. These short strands bind to the complementary sequences in the genetic code. RNAi works by stopping the information in the DNA from getting to the protein-making ribosomes. It interferes with the messenger RNA. When it comes to agricultural applications, RNAi can be used as a form of genetic pesticide that can be built right into a plant's biology. For example, an insect pest feeds on a crop that deploys RNAi coded to stop the ability for that pest to digest food and process nutrients from the plant. As a result, the pest's growth or its ability to reproduce is slowed or halted, or the pest dies. Or a crop produces RNAi that changes a plant's chemistry, making that plant unattractive to a pest. Or the RNAi can block a plant's susceptibility to an herbicide, allowing the herbicide a valuable asset in crop improvement and a powerful tool against yield loss. However, just as when using other powerful tools, we have to make sure that the technology is safe. Keep in mind that genes are made up of millions and billions of pairs of nucleotides, and RNAi targets gene sequences around 21 to 25 nucleotides long. That's right, 21 nucleotides in a sequence out of billions in a gene. With those kinds of odds, the chances of the RNAi blocking other RNAs from producing totally unpredicted proteins is likely. One possible risk is that the RNAi molecule might silence the correct gene, but in the wrong organism. It turns out that the RNAi and the RNA don't have to be 100% identical for there to be silencing. For example, in addition to silencing part of a corn pest digestive system, maybe an RNAi molecule would accidentally silence part of the digestive system of a lady beetle, or a honeybee, or a cow, or cousin Mabel. These risks are not trivial, especially because they're different from those posed by most other types of pesticides that we're used to dealing with. Now, considering the unintentional turning off of unintended genetic functions of targeted and non-targeted organisms, and the huge complexity of biological and ecological systems on which all life depends, this could be a bit of a problem. Without more knowledge about how pesticidal RNAi works in pests and non-target organisms, it's difficult to predict how this technology might affect the environment. Sounds dire, right? Well, it doesn't have to be. To manage these risks, we can already take some unknowns out of the equation to make sure that the pesticidal RNAi poses minimal threats to the species that we want to stick around. When you want to eliminate the unknown, what do you need? More data. You have to take steps to weed out RNAi that silences genes other than the one you mean to target, and be sure the genes they target are really involved in the cellular function of interest. So assessing an area's bioinventory can help. Then we'll know what species might be exposed to RNAi. Also, we'll get the genomes, or genetic blueprint, for the exposed species, and screen potential RNAi molecules to see whether a particular pesticide might hurt the species we want to keep healthy. From this, we can develop comprehensive risk assessment procedures that can make sure our desire to manage pests isn't coming at the expense of Mother Nature. Science can help us to understand the benefits and risks associated with this amazing new technology called RNAi, and where it fits in with a sustainable and long-term successful plan for agriculture.
For more information on...
All right. So RNAi has a application, right? People have definitely have used it in every possible sphere. For example, people with agriculture background have used it for agriculture, application. People with veterinary background have used it for veterinary uses, and people who are interested in the clinical findings are using it for understanding human diseases. Okay. It doesn't matter whether you're using it in animals, plants, or humans. The basic process is the same. The basic process has been understood in a lot of details, but applying it into the clinic, getting it into the clinic, or getting an RNAi engineered crop into your dining table is still a long way off. Okay, it is still a long way, because several, as we saw in this thing, several studies need to be done to show the safety of these genetically engineered products. Okay, so it is something that's time consuming by nature, but it is likely to have a potentially transformative effect. Okay, so I would like to stop at this point. Are there any questions? If there is any question, and if I know the answer, I will definitely let you know. Otherwise, I will definitely search for it and then let you know. Any questions? I think we should ask questions.
Yes, sir.
Yes. Go ahead.
Yes, sir. Am I on?
Yeah, you're right. Shakti, right? Go ahead.
Yes, sir. So, sir, I want to ask that can we stop the synthesis of protein by by adding or changing the codon to stop codon in the initial stage of a codon?
Sure. You can. The only thing is that you don't probably want to do that at the gene level, right? So people are interested in making it at the RNA level. But as I said, when it comes to monogenic diseases, people are more interested in correcting the error. Right now, the interest is in correcting the errors, right? So that is where we are. Why would you do that? Why would you add a stop codon to any gene? Like under what circumstances? I'm sure you have something in your mind. I am not catching it.
Yes, sir. So like I don't know, yesterday or the day before yesterday, you showed the cancer and the normal cell image, and there was one brown RNA in the cancer part.
Mm-hmm.
So
Very good, very
like
good.
yes, sir. So like, and in that case, can we stop RNA from expressing the protein by adding the stop codon in the
Right.
initial part?
Good, good, good. So if you look at that example, I just showed it for illustration purpose, but generally if you see the difference between a normal cell and a cancer cell, that brown RNA will be there in the normal cell also, but it's a very low amount, okay? But in a cancer cell, it will be overexpressed. Okay, so typically I use the example of cancer cells because that is something that is very well known. And as I said, it is not just one RNA. There are multiple, abnormalities in a cancer cell. So there are multiple messenger RNAs that are overexpressed or underexpressed. There are certain genes that are underexpressed in cancer cell. Okay, so it is a tough thing to do that at this point in time. But yes, probably when it comes to single gene diseases, if overexpression is causing an issue, it is possible that that could be edited. It could be edited. Okay.
Okay,
Good
thank you, sir.
question. Good question.
Thank you,
Okay.
sir.
Any other question? Okay. So keep thinking about it. If there are any questions, get back to me. All right. So we will stop the class and we will see tomorrow. Okay. Take care. God bless you all. Bye.
Thank you, sir.
Yeah.
Yes, sir.
Okay.